To do this by injecting LSD in mice and try to understand what is actually changing. And I am not going to introduce you all the story of psychedelics because Vicente had 1 hour to do it, and I think it was not enough. And I think one of the things that are very common on people that also work on psychedelics, at least we are very passionate about what we do, and we can talk for hours, weeks, months, years. So I will try also to make it short, but let's see how it goes. So I just wanted to introduce you to something that I call the Benjamin Button timeline of LSD. And this is going to be very briefly. But this is because I call it Benjamin Button because I don't know if you know the movie The Curious Case of Benjamin Button, which is the story about a man that is born as an adult and die as a baby. And I think, among others, research on LSD follow a similar path because due to the lack of legislation on drug prescription, it took only 4 years for the first clinical trial on LSD to be published after the first experience ever by Albert Hofmann, in comparison to more than 10 years for the first mouse study to be published. So back in the time, as Vicente also told you, LSD was given freely to all the psychiatrists and researchers to investigate what were these drugs were doing. And we already know the rest of the story. I will not take more time on that. Legislation, problems, and only now we are somehow restarting on the field of psychedelics. So today I want to try to give you some hints how we are studying this in the preclinical field about how LSD modifies the brain at all these levels, molecular, neuronal, and behavioral level, and also important at acute and more importantly at the long-term effects. So let's start with the basics, which is the receptor. If you know something about psychedelics in the field, probably you already heard about the 5-HT2A receptor. And these receptors, I will try to do an analogy for the non-scientific people in the house, but you need to imagine that LSD is kind of a key that will bind to this receptor that will be a door, okay? And then that will lead to specific pathways, specific behaviors, it doesn't matter now here. We know that at the acute level, LSD is binding to these receptors and it is producing acute effects. The most famous ones are the hallucinations, visual hallucinations, but it's not only reduced to that. And we also know this because we scientists have strategies to understand how this works. So, for example, we can put another key. And note that this key, so this key can fit the door, but it doesn't have the right shape to open it, but it blocks the door. So even if we have LSD, we cannot open this door. And you can do something else that is basically just remove the lock. So now even if you have the key, there is no lock to open the door. And for the scientists in the room, this I'm talking about the key. The first key would be the 5-HT2A receptor antagonists, and the lock of the door would be knockout studies of the 5-HT2A receptor gene. And also one thing that is very important is that we realize that there is also a correlation between the number of receptors that are occupied and the intensity of these acute effects. So this is one thing that we don't discuss too much in the field, that we kind of take for granted. And you're going to understand that there is not a lot of things that we can say for granted in the field. What we don't know, and I think what is also very important, is which specific pathways are responsible for the long-term outcomes. And now I'm going to show you a bunch of complicated metrics just for you to understand that this is very complicated. So don't try to understand everything. The idea is that you get confused. Okay? So
how many neuroscientists are there, by the way, just to... Okay.
We have half here.
Yeah, but also, feel free for questions and So even at these receptors that we know for sure that the psychedelics are interacting with, the 5-HT2A receptor, this is like a simplified view of what is going on. Okay, so it's much more complicated, and they also get into different proteins, depending on the dose. Like the overall thing is very, very complicated. And on top of that, let me tell you that from all psychedelics, LSD also has a lot of like what we call like a polypharmacology. So meaning it also opens a lot of different doors. Like imagine it as a master key that is not going to interact with the 5-HT2A. And I hope you can see that, and this is an incomplete list. Like it interacts with it actually interacts with 13 of the 15 human serotonergic receptors with agonist activity, antagonist with the other, and then it doesn't do nothing with one. We can discuss that after. All dopaminergic receptors, and now more recently adrenergic, histaminic receptors. It's a huge mess. In terms of neuroplasticity, one thing that we seem to know is that the psychedelics seem to promote neuroplasticity both in humans and rodents. So here is just like in yellow, you can see all human studies. These by the increase of BDNF protein, which is a proxy of an activation of the promotion of neuroplasticity. And in animals, we have a bunch of different studies. And what we can see is that actually psychedelics are going to increase genes that are related to neuroplasticity, that they are going to modify these neurons. So I hope you can see that if you compare the first cortical neuron here and the one that was treated with LSD, you can see that it got more complex, these are more synapses, more dendrites. So, and this is the kind of research we can do in the field. And one could argue that we have these metrics, but we don't know, for example, if these neurons are functional, because all of these studies are done in vitro, so not in an in vivo animal. For this, we also have studies at the functional level. So here I'm presenting you 3 papers. Again, this is just for you to believe in me of what I'm saying, but it's a paper, so papers on psilocybin, DOI, and LSD that actually show that maybe we have this increase of neuroplasticity that is also functional. One thing that we don't know is there a critical window for neuroplasticity? And what is causing this neuroplasticity? And this is like the interesting questions that we cannot address so well with humans. And I'm going to present you very briefly one paper that recently came out. It was published just 4 months ago. It comes from the Goodall Lab in Johns Hopkins University. And basically what they did was, imagine there is a mouse, imagine you are a mouse for now. You get an injection of a psychedelic or a saline as control. You became this mouse. Now seriously, you have the injection of the psychedelic here, okay? Then for 48 hours you go to your home where you have all your friends. And then after that, they do a pretest. So what they do is that they take you out of your home and they put you in a new environment. And then this environment, as you can see, it has like 2 colors here differently, but you don't know it. For you, it doesn't matter. We are just doing this to see as a control if you're going to prefer one of the environments. But for now, you don't have any reason to prefer to go to the blue or to the brownish environment. And after what the scientists do is that, okay, they say the blue environment now, your test 24 hours, and here you can be with one of your friends. Okay? Then after, in the yellow environment, you're staying there for 24 hours and you're alone. And then what they do is that they assess after 30 minutes which environment the animal prefers. And take into consideration that now the blue environment doesn't have friends, this is exactly the same as this environment was before, but you know now that the blue was associated with being with friends and the yellow not being with anyone. So they did this, and they modeled this for the animals with different ages, and they have this what they call the social preference score. So, meaning, if you would take more time in this environment, it means that you prefer or that to be with your little friends. And then if you would go more for this environment, it means that you would prefer somehow to be alone. And this gives you a score, and as you can see, there is some like these metrics here that depending on the age, you're going to have variable preference. So now you do this, but here, remember, you have the injection of a drug, okay? I'm going to talk about LSD because it's the subject of my thesis. So you give LSD, and what you see is that these are an animal that didn't receive any psychedelic, and when the animals receive LSD, their social score, so their preference to be in the space where they were, where is associated with being with their friends, goes really high. And then they did this by giving LSD 24 hours before. They're like, okay, this is quite interesting, what if we do it 2 weeks before? And they do this with a bunch of different psychedelics, and here you can see that both LSD and psilocybin made you prefer to be with your friends 2 weeks before. What if we do this 3 weeks before? Okay, psilocybin goes to normal, but LSD stays high. So what they concluded in this and what they presented, what I'm going to show you is not data, is a model of, like, is a cartoon to be easier to understand. But what they associated with this experiment was that in humans, the acute subjective effects also have different effects depending on the drugs. We know that ketamine, the acute effect is between 30 minutes and 2 hours. If you go to LSD, the acute effects go for 8 and 10 hours. And what they said is that there is a strong correlation and proportionality between the acute subjective effects in humans and the critical period of this social critical period open in mice. So they went even further with this to try to understand. Did you get this message? If not, you can ask questions and it's okay. But they went even further with this, and they tried to understand what is happening, and we have an hypothesis. So this is currently the most recent, and I would even say more, I don't want to say accepted hypothesis in the field, but the most recent hypothesis we have in the field, and it's only one paper, so take that also into consideration, that show that, but it's something that we can think about it, is that all of these drugs actually are going to acting through different receptors. Okay, here these are just name of the receptors. I try to put the colors so that you could associate with the little scheme here. They are going to do different things, as I told you at the beginning, that even with the 5-HT2A receptor, you had all these multiple pathways. But at the end, what is important in the hypothesis of these receptors is that you have a remodulation of these extracellular metrics, so this space here, that is going to be this permissive event that is going to allow plasticity to happen. They even go further and they talk about metaplasticity, which is basically all the changes that you need to have on the neurons to actually have the plasticity. So super simple put in words by Goudelin, the last author of this paper, she says it's the plasticity of the plasticity, and that's why we call call it metaplasticity. So again, just to show you that I hope you get a hint of maybe what's going on, but things are much more complicated again, and it's going to be always like this. In terms of neural circuits, what we know is that, one of the most famous images is these ones about the connectivity of the different brain regions. So here, imagine that all these little lobes are different regions in the brain, and when you give vehicle, you don't have a lot of connections, and when you have psilocybin, you have these representations. And here you have also, it's already a subtraction, so LSD minus vehicle. Imagine you would get this blob for LSD, you subtract this one, and you get the result. And the red just means increase of activity. So it means that this brain, it looks like it goes into this very connected state. And we can also assess this in mice. So Justine said that I could talk a bit about my PhD. One of the things I'm actually going to do is that I'm going to give psychedelics to the animals, and then I can extract their brains and try to understand what were the regions that are active by measuring proxies of neural activity. And it's what is trying to be represented here. So here, it's a paper that was published this year where they compared what are the brain regions that are active when you compare ketamine and psilocybin, and I just circle here the ones that are more active. It doesn't, you don't care about the names right now, okay? It's just like the brain regions that are more active on psilocybin. So we can have these metricals also in animal research. And another thing that is also very important to consider is that these effects are really dose dependent. I mean, I hope this is not new for you, but but these effects are really dose dependent. And here it's a paper that compares 3 different doses of LSD from 75 to 200. And it seems that the acute effects, depending on the dose, I hope you can understand that, or you can see, that the blue one that represents the higher dose of LSD, the score, it's increased compared to the others. In animal research, we can even go further, and this is just a paper to show you that with low doses of LSD, we are not affecting a region that has dopaminergic neurons, but that with high doses, we have a decrease of activity of dopaminergic neurons activity in the VTA, in this region. So all of this gets us closer to what is happening in the brain. But again, we don't know which modifications persist over time, and we don't know if the long-term effects are also dose-dependent. And there are different theories. I'm not going to go into details in any of them, but these different models in the field that propose an increase or decrease of different brain regions that might explain what is happening at an entire level in the brain. Then, in terms of behavior, psychedelics, as you know, become very famous because of the acute effects, because of hallucinations. Nowadays, they are still assessed in a more controlled manner because we still have the acute effects in the clinic. And so we can have this kind of metrics. Here is based on questionnaires where you ask people, you have standardized questionnaires, you ask people about their experience, and then you quantify them, again, depending on the dose. In rodents, let's see if I can show you. I will be able to show you. So in rodents, what's happening is that we also have an acute effect. So when we give psychedelics to mice, they have this response. You saw? So it's a very rapid movement with their head. I'm going to put it again. It's very cute. It's a whisking. It's not whisking. They move their head very, very rapidly. It's called the head-twitch response, and it's very, very characteristic for psychedelics. If there is one thing that we know in the animal research, in the rodents research, is that this is consistently happen with psychedelics in mice. And another thing that is quite interesting and cool is that there is also people that quantifies. I will tell you what the graph means. Again, this is for the scientists to believe me and what I'm saying. But it's basically depending on the potency of a drug to have an acute effect, you're going to have more a twitch response. Meaning, if I give you a drug and we know that this drug in humans is producing an acute effect that is with LSD that takes 8 hours, we are going to have the mouse doing this movement for a longer time. And this is a beautiful correlation that actually shows you that. At the long term, there are a lot of different metrics that we can also try to associate with human research and animal research. It's a bit a mess, to be honest. So I'm only going to talk about the ones that me as a researcher, Daniela, actually somehow trust in these studies, and not everything. So I'm going to give you 3 examples that, from what I understood from the literature, psychedelics can trigger sustained effects that are anxiolytic. So here, what you can see is that you have first, we put an animal in a box, we let the animal just move around the box, and usually mice, they don't like really to go to the center. They like to stay in the corners. Usually if they stay too much in the corners, it's because they are a bit anxious, or they have an anxious-like behavior. And they give it LSD and they see, okay, this animal is going more to the corners. There are statistics. You need to trust me on these. I can show you after. The antidepressive, it's based, I put it here, but it's based on also a controversial test in the field. So we don't have a lot of good metrics to evaluate antidepressive behavior in animals. What we do is that we basically put an animal in a little pool, and then we see how long it takes how it's swimming around. And if he's not swimming so much, we say that he has a depressive behavior. But this is a very controversial test in the field, but still it goes in the direction that with LSD, the animals are swimming more, so they are not so depressed somehow, or with depressed behavior. And then also about the prosocial, so I showed you this graph before, but there was also another paper that actually showed that. So this is somehow what we can, or the hints we have in the preclinical field to say what's happening in terms of the injury effects. But then we still remain with a super important question, that is, are the subjective effects of psychedelic necessary for the enduring ones? Does someone need to go through the psychedelic trip to actually have the positive outcomes, being a reduction in their depression symptoms or not having addiction? From the clinic, it suggests that there is a correlation. And one of the things that you need to know about science that we say all the time is that correlations are not causations, so be careful on that. But it seems that there is a correlation between the mystical experience, or the score that you give to your experience after taking the drug, and the reduction of your symptoms. And here also, again, you can see this one, I put it because it's on LSD, the responders and non-responders. It seems that the responders, they went through a more experience, like intense experience. From the preclinical field, what we can do is actually we can modify these drugs, and now they don't produce the acute effect. And I'm going to show you 3 examples of when they did that and what happened. So they modified the cocaine, and there was still a reduction. Okay, this is a reduction on alcohol intake with the drug, that is similar to ibogaine but is not producing the acute effects. And there is not because the animals are not drinking so much. Again, with the swimming test, there was a modification of a 5-HT2A receptor agonist to be very specific. And they also show that with this modification, so you see that there is these compounds. Okay, here, you need to look for this color only. And there was a reduction from day 0 to day 14, at least. And again, this is a drug that is not producing the acute effects and still is having increase in the mobility of the animals that is then associated with this antidepressive-like behavior. And here is just a scheme, but again what they did is that they modified LSD to bromo-LSD. So if you modify it not to have the acute effects, the animal is not going to do this lethal behavior I show you, but they showed that it could reverse chronic stress effects and induce structural plasticity. So all of these results also I put into the question and into the discussion if it's necessary to have this psychedelic-like experience to actually have the outcome effects. Right now, we don't have any of these drugs approved to human use, so we don't know if this would stand also for human research. And I was actually very brief, guys, but I want to discuss with you. So I think I show you very briefly that in terms of neuroplasticity, receptors, neural circuits, and behavior, this is more or less what we know. Most of the research that we have are on the acute effects when the most interesting results, I think in my opinion and in general opinion, is actually the long-term effects and how these drugs can help really people with disorders. And I just want to say thank you. I did this, Dali did this image on the prompt of a woman neuroscientist standing on the shoulders of giants in a psychedelic style. And I just want to thank everyone that was represented here, all the scientists that were represented here, all the animals, all of you for listening, and the organizers for inviting me, because I love to talk about my research. It's the first time I'm doing for a public that is not scientific, so I'm sorry if I was a bit too broad or not specific, but we can discuss more details, and I hope you could get an overview of what we are doing in the animal research field.
Nice, thank you!